हेलो गाइस। तो आज हम लोग KCET 2026 का बायोलॉजी का पेपर डिस्कस करने वाले हैं। तो मेरे पास ये जो पेपर है ये A3 वाला है, यानी कि A3 कोड मेरे पास आया है। तो आप लोगों के पास जो भी कोड हो उसके अकॉर्डिंग आप अपने क्वेश्चंस को देख सकते हैं और हम लोग यहाँ पर पूरे के पूरे 60 क्वेश्चंस को अच्छी तरीके से सॉल्व करेंगे और हो सका तो उनके कंसेप्ट को रिवाइज भी करेंगे। तो चलिए शुरू करते हैं। तो जैसा कि आप सब लोग देख सकते हैं A3 के अंदर जो सबसे पहला क्वेश्चन है आप लोगों के सामने, विच वन ऑफ द फॉलोइंग ऑप्शंस रिप्रेजेंट्स द स्टेप्स ऑफ डीकम्पोजिशन इन सीक्वेंस। ठीक है, तो अब ये जो क्वेश्चन है ये इकोलॉजी वाले जो हमारा यूनिट है वहाँ से है। अगर इसके अंदर हम ध्यान से देखें तो आप लोगों को ऑप्शंस में दे रखा है फ्रेगमेंटेशन, फिर उसके बाद आपको दे रखा है लीचिंग, कैटाबोलिज्म, ह्यूमिफिकेशन और मिनरलाइजेशन। तो इसमें जो करेक्ट ऑप्शन होगा वो हमारा सेकंड होगा। क्यों? क्योंकि देखो अगर कोई भी चीज़ डिकम्पोज होना स्टार्ट होती है तो ये चीज़ एनसीईआरटी में क्लियर कट मेंशन की रखी है कि सबसे पहले वो छोटे छोटे पार्ट्स में टूटेगी, यानी कि उसका फ्रेगमेंटेशन होगा, उसके फ्रेगमेंट्स होंगे। जैसे ही फ्रेगमेंट्स कंप्लीट हो जाते हैं उसके बाद होती है लीचिंग। लीचिंग का मतलब होता है कि जो छोटे छोटे पर्टिकुलर पार्ट्स हैं ना वो रिस के नीचे की तरफ आते हैं और माइक्रो ऑर्गेनिजम्स के कॉन्टैक्ट में आते हैं। देन उसके बाद होता है कैटाबोलिज्म। तो कैटाबोलिज्म अगर आपको याद हो तो ये एक ऐसी मेटाबॉलिक रिएक्शन होती है जिसमें कॉम्प्लेक्स ऑर्गेनिक मॉलिक्यूल ब्रेक डाउन होता है सिंपल इनऑर्गेनिक मॉलिक्यूल्स के अंदर, राइट। सो देन आफ्टर ह्यूमिफिकेशन इज द प्रोसेस ऑफ फॉर्मेशन ऑफ ह्यूमस। नाउ व्हाट इज ह्यूमस? ह्यूमस इज द डार्क ऑर्गेनिक सब्सटेंसेस जो कि फॉर्म होता है आफ्टर डिकम्पोजिशन ऑफ ऑर्गेनिक मैटर। एंड द ह्यूमस एक तरीके की एक्सोथर्मिक रिएक्शन से बनता है। सो ह्यूमस इज द ऑर्गेनिक मैटर विच ऑल्सो यूज्ड एज मैन्योर। सो लास्ट प्रोसेस हमारे पास आ जाएगा मिनरलाइजेशन। मिनरलाइजेशन का मतलब होता है कि जो भी सब्सटेंसेज हैं, ऑल दो सब्सटेंसेज आर कन्वर्ट इनटू द मिनरल्स, लाइक फ्रॉम ऑर्गेनिक टू इनऑर्गेनिक मिनरल्स। ओके, सो जो हमारे पास ऑप्शन हो जाएगा वो हो जाएगा सेकंड। नेक्स्ट हमारे पास है दी नेट प्राइमरी प्रोडक्टिविटी ऑन लैंड एंड ओशन रिस्पेक्टिवली आर। तो गाइज वैसे तो 70% ओशन है हमारे पास और 30% ऑलमोस्ट लैंड है अगर हम एरिया की बात करें, सरफेस एरिया की। लेकिन ये एनसीईआरटी के अंदर साफ साफ मेंशन है कि लैंड का जो नेट प्राइमरी प्रोडक्टिविटी है वो ओशन से कहीं गुना ज़्यादा है, राइट। तो आपका आंसर हो जाएगा 115 एंड 55, यानी कि आंसर आपका आ जाएगा इसका सेकेंड। नेक्स्ट उसके बाद हम हमारा क्वेश्चन देखते हैं। तो नेक्स्ट क्वेश्चन जो है ये पेडिग्री एनालिसिस का है। पेडिग्री एनालिसिस जो है वो बहुत ही ज़्यादा मुश्किल लगता है बच्चों को, बट ये होता नहीं है। इफ़ यू नो हाउ टू डू दी मोनोहाइब्रिड क्रॉस और डायहाइब्रिड क्रॉस देन इट इज़ वेरी इजी टू अंडरस्टैंड। सो दिस पेडिग्री, अगर हम इस पेडिग्री के बारे में बात करें तो दिस पेडिग्री इज डायरेक्टली टेकन अप फ्रॉम द NCERT चैप्टर नंबर 4 एंड वहाँ पर ये मायोटोनिक डिस्ट्रॉपी की पेडिग्री बताई गई थी। और जो मायोटोनिक डिस्ट्रॉपी है दैट इज ऑटोजोमल डोमिनेंट, ये ऑटोजोमल डोमिनेंट की पेडिग्री है। नाउ व्हाट इज द मीनिंग ऑफ ऑटोजोमल डोमिनेंट? व्हाई? बिकॉज यू कैन सी मेल एंड फीमेल आर इक्वली अफेक्टेड, so it's not the sex linked dominant or recessive. And another thing is, if you can see, if it is an autosomal dominant, that means those individuals which are not affected must have genotype small a, right? And the person who is affected can have genotype capital A small a or capital A capital A. So like this we can say, अगर ये वाला कंडीशन होता तो सारे के सारे जो बच्चे होते उनके अंदर इस तरीके की प्रॉब्लम होती, राइट। बट ऐसा तो नहीं हुआ है। इसका मतलब यह है कि यहां पर दिस इज द कंसेप्ट, दिस इज द जीनोटाइप, राइट। एंड दिस जीनोटाइप, यहां पर जो भी आपको डिजीज दिखाई दे रहा है उनके पास जीनोटाइप होगा कैपिटल ए स्मॉल ए, एंड दोज विच आर नॉट अफेक्टेड मस्ट हैव द जीनोटाइप स्मॉल ए। So here you can see the small a genotype of male and the female partner also have small a genotype. So you can see all the progenies in the third generation are not affected because none of the parents are affected, right. And here this is the genotype of this parent, this may be the genotype of this parent. So all these are small a and this one can be the heterozygous condition, right. So this is the pedigree of autosomal dominant which is given in the NCERT for myotonic dystrophy. Let's go to the next question. The next question is, given below are two statements. Statement 1, there is a less species biodiversity in tropical latitudes. Now, tropical latitudes, this is not the case. Tropical latitudes does not have less biodiversity as compared to temperate region. The tropical region is the middle one where evergreen forests are there. The temperate region is slightly upper and lower portion, right? So, the tropical region, if you can see the next statement, the tropical environments unlike temperate one are less seasonal, relatively more constant and predictable. Tropical regions, because they are evergreen forest, so there is no condition of drought or something like that. And because of that, the temperature or the climate remains constant, and this constant temperature give chance to the other animals and plants to grow more in that area. The condition or any place where the climate is fluctuated, there can be the conditions where animals can't grow properly, है ना भाई? सिंपल सी बात है, अगर आप अपने घर में हो और घर में सेम तरीके का माहौल है तो आपको पता है, प्रिडिक्टेबल है कि आप क्या होने वाला है. लेकिन अगर आप किसी ऐसी जगह पे हो जहाँ पे पता ही नहीं है कब बारिश हो जाए, कब धूप निकल आए, तो फिर वहाँ पे सर्वाइव करना थोड़ा सा मुश्किल हो पाता है, राइट? सो दिस इज द wrong statement because it says less species diversity in tropical latitudes. Actually tropical latitudes में more species diversity होती है और temperate के अंदर less होती है, temperate के अंदर less होती है. So this statement is wrong, but this statement is correct. So our option, correct option will be the statement 1 is false, but the statement 2 is true, right? Let's go to the next. Now here you can see, आपको फ्लोरल फॉर्मूला पता लगाना है और यहाँ पर आपको काफ़ी सारे ऐसे स्पेसिफिकेशंस और कैरेक्टर्स दे रखे हैं जिनके थ्रू आप करेक्ट फ्लोरल फॉर्मूला को पहचान सकते हैं. सो दिस मस्ट बी बाइसेक्सुअल, बाइसेक्सुअल इज दिस वन, राइट? एक्टिनोमॉर्फिक मीन्स दिस वन, it can be divided into two equal identical half for more than one plane, right? Pentamerous, pentamerous condition means the petals and sepals are in the set of 5, 10, 15, right? So K must be 5, C must be 5, and they are gamosepalous and gamopetalous. That means we can put a bracket on them, right? After that, 5 stamens, that means androeciums are 5. Epipetalous condition, epipetalous condition means the filament of androecium is attached with the petal, right? So how do we show it? Like this, right? And bicarpellary, it means two carpels are there, syncarpus, that means they are fused, and superior ovary, that means hypogynous condition, right? So the correct formula according to this will be the fourth one, right? Okay, let's come into the next question. So question number 6 says, a student asked to identify a permanent slide of mitosis, he made the following observation. Okay, mitosis की slide है and the observations are splitting of centromere and formation of daughter chromatid. Now you know that when we are talking about these slides of mitosis or the phases of mitosis, in prophase, nuclear membrane starts to disappear and chromatid condense to form the chromosomes, right? In metaphase, they arrange on this equator site like this, and in anaphase, the centromere splits and we got this kind of condition, like this kind of condition. And the telomere or telophase is the opposite of prophase, just opposite of prophase, right? So splitting of centromere, this is the characteristic feature of mitosis. Splitting of centromere ensures that equal number of chromosomes goes in the both daughter cells, right? And formation of daughter chromatids, and chromatids are migrating towards the opposite pole. तो chromatids ने opposite pole की तरफ migrate करना start कर दिया है. So our correct option will be third anaphase. It is a direct question, right? Class 11th chapter cell cycle. Okay. The next question is, with respect to binomial nomenclature, the binomial nomenclature is given by Carl von Linné and he उसने अपना नाम भी Carl von Linné से Carolus Linnaeus में convert कर लिया था. Okay, so with respect to binomial nomenclature, identify the incorrect statement. What we have to find is, we have to find the incorrect statement, right? So biological names are generally in Latin and written in italics. Okay, now this statement might be wrong. Why? Because it's not written in italics, it's typed in italics. When we write them, we separately underline them. Okay, this is the genus name, this is the specific epithet, species name. So when we handwritten, because some people write in italics, that's their normal, you can say the way of writing, है ना. बहुत सारे लोग लिखते हैं जैसे हवा चल जाती है ना, सारे के सारे शब्द उनके टेढ़े हो जाते हैं, right? So many people write in italics. So it's not the written in italics, it must be the typed in italics. But let's see the other options also. The first word in the name represents specific epithet. Okay, that is the completely wrong statement. First word always represent the genus and the second word always represent the specific epithet. Okay, so this is completely wrong, and with respect to this one we can say this can be right. Okay, both the words in the name when handwritten are separately underlined. See, when handwritten separately underlined, right? This is the right statement, and the first word starts with the capital letter while the second word starts with the small letter. This is also the right statement. Okay, so the most incorrect one must be this second. Okay, the answer will be the second one, right? Okay, so let's go to the next question, question number 8. The organism X is multicellular. Okay, if it is multicellular, that must be, it is not the moneron and it is not the protestant. It can be fungi, it can be animal, it can be plant. Heterotropic. Okay, now heterotropic means it can be animal or it can be fungi. Plants जो है यहाँ से निकल गए. Eukaryote. Okay, both of them are eukaryote. Chitinous cell wall. Now this is the deciding factor. Chitinous cell wall. अगर ये सारी चीजें नहीं भी दी होती, अगर ये सारी चीजें नहीं भी दी होती, with only this character we can say that the organism is fungi. Okay, so the organism which have chitinous cell wall, the cell wall made up of chitin, must be placed under the fungi kingdom, right? So coming to the next question, question number 9. So read the following statement about funaria. Now the funaria is a bryophyte. Okay, and this is not liverwort. In bryophyte also funaria is a moss, right? Funaria is a moss. Okay, so select the options which are correct. Now we have to choose the correct option. We have to choose the correct option, right? The gametophyte is a dominant plant body. This is the correct statement, right? The sporophyte is differentiated into foot, seta and capsule. This is the correct statement. Sporophyte have foot, seta and capsule, right? The gamma are asexual, green and multicellular buds. This is completely good, okay? And found on thallus. This is also right, but this is right in terms of liverworts like Markensia. This is not true for funaria. This is not true for funaria. Funaria, that means moss, they have asexual reproduction through protonema formation. Okay, अगर आप इसको पढ़ेंगे क्लास 11th चैप्टर नंबर 3 प्लांट किंगडम, तो उसके अंदर आपको पता चलेगा कि फ्यूनेरिया इज अ टाइप ऑफ लिवर मॉस, एंड इन द मॉसेस दे हैव द asexual reproduction done by protonema formation, while in the liverworts they have gamma cups present on their thallus, and from that gamma cup, gamma are produced which are multicellular, green and photosynthetic asexual buds, right? So this is not correct with respect to funaria. The statement is correct but not with respect to funaria. Okay, the sporophyte has independent existence. Now this is wrong, this is also wrong. In all the bryophytes, sporophyte is dependent on the gametophyte body. Okay, sporophyte is not photosynthetic, it acquires its nutrition from the gametophyte thallus, right? So these C and D are wrong. That means A and B are right. So the correct option must be the first one, right? Okay. Let's go to the next 10th. Match the following. Okay. So we have list 1, we have list 2. If we can say the excretory organ from list 1 and list 2, this question is from the animal kingdom, chapter number 4, class 11th. Leech is a member of Annelida, right? Leech is the only member of Annelida with open circulatory system, but they have what? Nephridia. They have what? Nephridia as their excretory organs. So we can see that A is third for only this option. So this option must be correct, but we can check the other one. Locust is an insect. Okay, locust is an insect, and insect are you can say the main or sole member, not the sole member, but the main member of Orthopoda, the largest phylum of animal kingdom. Now the insects are those which have 6 feet, 3 pair of feet, like cockroach, like flies, like mosquitoes, and they have Malpighian tubules. Cockroach is also in your course, so they have Malpighian tubules as their excretory organs. So locusts have Malpighian tubules. Other arthropods have book gills, book lungs, these kind of things. Liver fluke. Now liver fluke is the member of Platyhelminthes, that is flatworms, and the flatworms have flame cells. Flatworm have flame cells as their excretory organs. While the Balanoglossus is from Hemichordata and it have proboscis gland as its excretory system. So you can see the third option is matches correctly with the everything, right? Okay. So let's go to the next question. Question number 11. Identify the flower with polyadelphous condition. Now polyadelphous condition means different groups of androecium, and this can be seen in citrus, not in mustard, not in China rose, not even in pea. In pea we have 2 different groups, 9 are together, one is alone, but this the alone one is not considered as group. Okay, it is a single stamen. Okay, if you can see, the pea have this kind of estivation, and 1, 2, 3, 4, 5, 6, 7, 8, 9 and 1, this kind of conditions in respect to stem. But the polyadelphous is citrus, it have many androeciums. Okay. Second, 12th question. The large empty colorless cells is seen in upper epidermis of grasses which facilitate the curling of leaves to minimize the water loss. Now this is the direct question. The epidermis have some bigger, like this, colorless cells which can, like this. Okay, these are like this. Now in the condition of water scarcity, these cells, also known as bulliform cells, they reduced in size due to loss of water, and as they reduced in size, the leaf starts to curl like this, like this. Okay, so stomatas all are covered and the water loss can be prohibited, right? So these are known as bulliform cells. The second one is the correct answer. Direct question from the anatomy of plants. So let's go to the next question, the question number 13. So find the incorrect statement among the following with respect to the digestive system of frog. Now we have to consider a statement which is incorrect with respect to frog, right? So food is captured by bilobed tongue. This is completely true. The frog have bilobed tongue, है ना? आपने देखा भी होगा वो इस तरीके से जीभ निकालता है और पकड़ लेता है इंसेक्ट को, right? The esophagus is long tube. Now this is incorrect. The esophagus is very short in frog and opens into the stomach. This is correct thing, and continues as intestine. This is the correct thing. But here the long tube is the wrong one. So this is the wrong statement. Liver secrete bile that is stored in gallbladder. This is the correct statement. and the undigested solid waste moves into the rectum and thus passes through the cloaca. Now you know that the cloaca is the common opening for reproductive system in frog, excretory system as well as defecation, right? So cloaca is the common opening, so this statement is also correct. So the correct answer from this will be second, right? Second is the incorrect one. Going to the next question, question number 14. Which of the following is not the function of plasma membrane? Now, plasma membrane can do endocytosis, that is cell drinking or cell eating process. Endocytosis is the cell eating process, while pinocytosis is the cell drinking process. Formation of intracellular junction, this is completely correct. This kind of intracellular junctions are formed by plasma membrane, right? Secretion, this is also correct. Plasma membrane can secrete something. What happens when we have the cells which can secrete something? They are termed as glands. So glands must produce something and secrete. Now the plasma membrane is the outermost covering, so it must have function of secretion, right? But DNA synthesis, now DNA synthesis is not a function of plasma membrane. So the correct option is fourth. Okay. So these are very easy questions. If you can see, they are completely NCERT based. So let's come to the question number 15. A chromosome with an extremely short and a very long arm is called. Now many students think this is a telocentric chromosome, but it is not a telocentric chromosome. The NCERT says the telocentric chromosome only have one arm because it have terminal centromere, right? And it is clearly written in the NCERT that some chromosome with extremely short and very long arm must be the acrocentric chromosome. The acrocentric chromosome are like this one. They are also called as J-shape chromosomes, right? So moving on to the next 16th question. So the following statements are the steps in the catalytic action of enzyme. Arrange them the correct sequence. So catalytic action of enzyme means how enzyme works and how enzyme convert substrate into the product. That is called the turnover number. Okay. So we have to correct, arrange them in the correct sequence. Okay. So let's first understand what are the statements. The active site of enzyme is now in close proximity of the substrate, breaks the chemical bonds of the substrate and new enzyme product complex is formed. So here enzyme product complex is formed already, but this can't be the first step because first enzyme comes near the substrate, then enzyme substrate complex formed, then enzyme product complex formed, and then enzymes plus product, right? This is the equation which is written in your NCERT. So the second statement is the substrate binds tightly to the enzyme, inducing change of active site of the enzyme. Now this is the correct statement. When the substrate bind with the active site, a slight change in size and the shape of active site is seen in the enzyme, and this is the particular process which is required for tight binding and change in the chemical properties or bond of the substrate, right? Third, the enzyme releases product. So this must be the last step. This enzyme also already have released the product. Okay. And the substrate bind to the active site of the enzyme. Now, substrate bind to the active site. This must be the first one. Okay. First enzyme bind with substrate. Right. After that, what happens is the C1 where enzyme already produced the product, this must be the last one, right? The substrate bind tightly with the enzyme. So when the substrate bind with the active site, the second must be the substrate bind tightly with the enzyme. The third one must be the active site of enzyme now in close proximity, and the chemical changes happen. And the fourth step must be the enzyme release the product, right? So the correct option must be DBAC, right? Okay. So let's go to the next question, the question 17. A diploid cell, just a moment, yeah, a diploid cell which have 8 chromosomes undergoes meiosis. Now meiosis is reductional division, that means the chromosome numbers must be half in the daughter cell, right? What is the chromosome present in each daughter cell? Now this is a very easy question. If the parent cell have 8 chromosomes, then the daughter cells formed must have 4 chromosomes, right? This is the direct question. The correct option is the second one in this case. Cellulose is the important structural component of the plant which is made up of, this is glucose. Cellulose is the polymer of glucose. Yes, fat is also the polymer of glucose, starch is also the polymer of glucose, as well as cellulose is also a polymer of glucose. The only difference is cellulose have beta 1,6 linkage. It's like this. This is first glucose, this is second glucose, this is third glucose, this is fourth glucose, this is fifth glucose, and so on. So this kind of linkage is present in the cellulose. Okay. Let's go to the next question. A healthy individual, the glomerular filtration rate, GFR. Now GFR is 180 liters per day. That is also 125 ml per minute. Now this is the direct question, it is given in the NCERT. 125 ml per minute. Next, a hormone that initiates flowering and synchronizing fruit set in pineapples. If it is only flowering, we can go for auxin or gibberellic. But the synchronized fruit set. Now why synchronized fruit set? Pineapple is a fruit which is made up of whole inflorescence, not from a single flower. It is made up of whole inflorescence, right? So it's like this. There are many flowers like this. There are many flowers. These are different flowers. Now every flower must change into the fruit simultaneously, है ना? That is not possible कि आधे flower तो fruit में change हो गए और आधा pineapple flower में रह गया. ये possible नहीं है, right? So all the flowers must change into the fruit simultaneously, and this is known as synchronizing. That means the change is occurring at the same time, and this can be done by ethylene. This can be done by ethylene. The correct option is first one, ethylene. Let's go to the 21st chromosome. Identify the incorrect statement regarding the respiratory system in man. Respiratory system in man. Okay. So lungs are covered by double layer membrane called pleura. This is the correct one. Lungs have double membrane. Yeah. Parietal pleura and visceral pleura. These two are the membranes, right? The alveoli are surrounded by rich blood capillaries. This is also correct statement, because alveoli are the site of gases exchange. So the oxygen and carbon dioxide must exchange from blood to the alveoli at this particular point, and for that alveoli must be in the close proximity of very fine capillaries, right? So this is the correct statement. The trachea, bronchi and bronchioles are supported by O shape. Now this is completely wrong. They are C-shaped cartilaginous ring. C-shaped cartilaginous ring. So this is the correct incorrect statement. The right lung is slightly larger than the left lung. This is also correct, because the left lung have cardiac notch, the space which is occupied by heart. Okay, so right lung is slightly larger than the left lung. So these three are correct, and we have to identify the incorrect statement, and third one is the incorrect statement. So the option third is the correct option for this question. Okay, now why C shape, not O shape? See, cartilage is the cartilage which is present here in our trachea. It is hyaline cartilage, and it provides the rigidity to the our windpipe or trachea, so that it cannot collapse. Okay, if we are forcefully expire the air, the pressure inside can decrease, and the pressure which is present in nearby the trachea can push it and collapse the trachea. These C-shaped cartilaginous rings act as their exoskeleton, right? Act as their exoskeleton, and they protect the trachea from collapsing. Okay, I am saying exoskeleton in terms of trachea, not for in terms of us. Okay, this is completely same as umbrella. The umbrella have spikes which are made up of metal. If we remove all the spikes, the umbrella is nothing but a cloth, so it will fall down, right? So the same concept is here. Now if this is the case, it must be the complete O shape. It will give the more stability to the trachea, isn't it? Now it is C shape. Why? Because it's like this. This is our windpipe and this is the C-shaped cartilaginous ring, and here is our food pipe, right? If it is complete O shape, then when peristalsis occurs in the food pipe, our food pipe cell can be damaged because the cartilaginous ring is made up of cartilage, which is a tough tissue, right? So to prevent it, the nature smartly placed it like a C shape. So from here it is completely protected, and here a small portion is left out so that the peristalsis can be easily done in the food pipe, right? Okay. Going to the next 22nd question. Consider the following statement with respect to ECG, electrocardiogram, and choose the correct answer. We have to choose the correct answer. Okay. So the P wave represent depolarization of ventricle. Now this is incorrect. If we can draw it, it's like this is the P wave, this is the QRS complex, sorry. This is the P wave, this is the QRS complex, and this is the T wave, right? P, QRS complex and T wave. Now the P wave represents the polarization of atrium. It has nothing to do with ventricles. So this is the incorrect statement. Now the statement second says T wave represents the repolarization of ventricle. Now T wave represents joint diastole. If you can recall from the cardiac cycle, 0.8 second is the duration of one cardiac cycle, in which 0.1 second is atrial systole and then 0.7 second is atrial diastole, and the ventricle systole for 0.3 seconds and rest 0.5 seconds are their diastolic process. So 0.7 second for atrium, 0.5 second for ventricle. In this case, 0.4 second is called as joint diastole. Joint diastole means they are relaxing again, and if they are relaxing again, that means they are going in repolarization. They are going in repolarization so that they can again do the contraction, right? So this is the correct statement. So the statement 1 is wrong and the statement second is correct. So our correct option will be third one, right? So let's go to the next. Aldosterone from the adrenal cortex stimulates the reabsorption of sodium, okay, from proximal convoluted tubule, ascending limb of Hamlet, distal convoluted tubule or descending loop of Hamlet. Now the NCERT clearly states that if there is a conditional reabsorption or secretion, that means there is a reabsorption and secretion in the normal time, okay, when we are not in the water scarcity or something like that. But if we have to absorb more water because we have water scarcity in our body, we are dehydrated, then we need to pass the concentrated urine, right? In order to pass the concentrated urine, there are some systems work in our body like RAS system or ADH. Aldosterone is the participant of the RAS system, renin angiotensin aldosterone system, okay. Now it is completely written in the NCERT that the conditional reabsorption is always done in DCT or collecting duct. That means later part of the nephron. Why? Because PCT have fixed reabsorption pattern. 100% glucose, 100% amino acid, almost 70% of the water, almost 70% of the salt. So PCT does not change. जो भी change है, जो भी गड़बड़ घोटाला है, that is done in the later tubular part of the nephron, which represents DCT. So our correct option is DCT, distal convoluted tubule. It doesn't matter if we are talking about ADH, it doesn't matter if we are talking about RAS system. If it is conditional reabsorption, it will be always done by DCT. Okay. So next question from the locomotion and movement. So this is the complete chart, we have to fill it. Okay, fibrous joint. Fibrous joint is present in the cranial sutures. Okay, there are 8 bones in our complete cranium: frontal, parietal, temporal and occipital. And all these bones are completely fused together. We cannot move them, right? We cannot move them. This is called as cranial sutures. Now B, between adjacent vertebrae, this provide a little bit of motion and they are cartilaginous joint. They are cartilaginous joints, right? And synovial are the type of joint which provide whole degree of motion. Okay, so the C is present between atlas and axis vertebra. Now what is atlas and axis vertebra? We have 7 cervical vertebra in our neck. Okay, from here to here, we have 7 cervical vertebra. The first cervical vertebra is known as atlas and it is like something this, something like this, and it has a hole in between, right? Now our skull is dicondylic, means we have 2 condyles which are present in the occipital bone, and our skull is present like this on the atlas. Okay, now this second bone is axis, which have this kind of odontoid process. It is also called as odontoid process, and this process fit here like this. This process fit here. So this is like odontoid process and this is the central canal of the atlas here like this. So this kind of motion, no motion. When we say no, we use this joint, left and right. So what is this joint which is present in the atlas and axis vertebra? This is pivot joint. This is pivot joint, right? And hinge joint means only motion in this direction, only one direction. I can't move in the another direction. So our elbow joint, our knee joint are the examples of hinge joint. So elbow or knee, right? So A must be cranial sutures, yeah. This is the correct option, right? Okay, coming to the next question. The unipolar neurons. Now what are the unipolar neurons? Like on the basis of projection, we have different kind of neurons. Okay, first is apolar. Apolar neurons only have cyton. They don't have dendrites, they don't have axons. Unipolar are those which have only axon, no dendrites, one cyton and one axon. Bipolar are those which have one axon and one dendrite, one dendron, like this. And the multipolars are those which have one cyton, one axon and many dendrites, These kind of neurons are present in our cerebral cortex, in our brain, spinal cord. This kind of neurons are present in the sense organ like retina of our eye. Now this kind of neurons are present in the embryonic stage when we are embryo. Okay, and these kind of neurons are not present in the human beings. They are found in very primitive organisms like hydra, coelenterates. Okay, so unipolar neurons are found usually in embryonic stage and they have one cell body and one axon. This is the correct option. Okay, so let's go to the next. Gastrin is the hormone secreted by GI tract which stimulates the secretion of hydrochloric acid and pepsinogen. So gastrin is an enzyme which is secreted by the GIT tract of stomach and it works on stomach. Okay, it is secreted by stomach and works on stomach. So what are the secretions of stomach? Main secretion, hydrochloric acid and pepsinogen, right? So this is the correct option, option number 1. Let's go to the next question. Choose the correct sequence of wall layers in a microsporangium microsporangium of angiosperm means the young anther. We are talking about young anther. Okay, so young anther have outer epidermis, inner one is endothecium. Both of these layer are single layer. Okay, then multi-layer middle layer and one tapetum which have syncytium condition, more than one nucleus, right? So epidermis, endothecium, middle layer, tapetum. This is the correct option, right, from outer to inner. Let's go to the next question, question number 28 here. Which of the following statement is correct? Correct statement we have to find for clistogamous flower. Now, what are clistogamous flower? In the first chapter, sexual reproduction in flowering plant, we have the examples of viola, oxalis. They have two type of flower. One is chasmogamous. Chasmogamous means open flower. Okay, the flower which we are used to see. Okay, and the clisto. Clist means fist, and we can only make a fist after closing our fingers. Like so, this is the closed flower. This is the closed flower, clistogamous flower. So they have exposed anther and stigma. This is completely wrong. Chasmogamous flower have open stigma and anther. Clistogamous flower means they are completely closed, right? They produce a short seedless set. Now this is the correct option. Why? Because doesn't matter if the pollinators are there, doesn't matter if the pollen grains are produced somewhere else or it will be transferred or not. If it is a closed flower, that means it is the bisexual flower and it will surely produce a seed. Why? Because it does not depends on any kind of pollinators. So it is the assured seed set. They need pollinators for pollination? No, because the flower is completely closed. The pollinators can't enter it. Okay. And variation of trait are not seen in the next generation. Now this is also the correct one. Why? Because the closed flower, the genetics are same in the pollen tube or male gamete formation, pollen grain and male gamete formation, as well as in the embryo sac and egg cell, because it is made up of same flower, right? So the same genetics are fusing together and forming a zygote. Now if the same genetics are present there, that means there is no mixing of any foreign DNA, and if there is no foreign DNA, that means there is no variation, right? So B and D are the correct one. So statement B and D are the correct one. This one, third, is the correct option for us, right? So let's go another one. Identify the drug molecule from the skeletal structure shown below. This is the direct question. These are cannabinoids. These are the chemicals which are made from the charas, ganja, or these kind of alkaloids. These kind of compounds have cannabinoids. Okay, and they attack or they affect on our central nervous system, right? So this is the direct question. The skeleton is present in the NCERT, right? Next, identify the correct path of milk secretion by mammary glands. Milk secretion by mammary glands. So mammary gland, like this, they have alveoli where milk formation occurs. Then they have small alveolar tubules. Many small alveolar tubules combine to form alveolar duct. They are enter into the region which is called as ampulla, and after that lactiferous duct is formed, right? So it is alveoli, mammary duct. Now this is wrong. Alveoli, mammary tubule, mammary duct, ampulla, and lactiferous duct. This is the correct statement, right? This is the correct sequence. Okay, let's go to the next question. We have to match the column 1 from column 2. So the acrosome head, middle piece of the tail. So these all are the parts of a human sperm, right? Acrosome is the portion which is filled with the enzyme that help in fertilization, right? Hyaluronidase enzyme and some other enzymes. Now head is the portion which contains mitochondria. No. Elongated haploid nucleus. Middle piece contains many mitochondria, and the tail is responsible for the sperm motility. So the correct option must be A third, B fourth. A third, B fourth. This is one. This one is the correct option. Okay. Let's go to the next question. Which of the following does not produce seminal plasma? Now, what is seminal plasma? Seminal plasma is the 90% content of semen. Semen is made up of seminal plasma and sperms. Okay. So, seminal plasma is the secretion of three reproductive glands, that is seminal vesicle, prostate gland and bulbourethral gland, which is also known as Cowper's gland. So, epididymis is the portion which is present near testis, over testis, right? It is highly coiled tubular structure, have caput, corpus, cauda, head, body and tail. These three are the main parts of the epididymis, and epididymis temporarily stores the sperm and also functional and we can say physical maturity happens in the epididymis. But epididymis does not produce any kind of seminal plasma or any constituent for seminal plasma. Okay. So, these three are glands, bulbourethral, seminal vesicle, these three are glands. So, not produce seminal plasma is the epididymis. Direct question. Okay. Let's go to the next question. Which of the following STD is not completely curable? So, first of all, you know AIDS is not completely curable, right? Genital warts are curable. Chlamydiasis and gonorrhea, these are of a bacterial disease, they are also curable. Genital warts and hepatitis B. Now, hepatitis B is not curable, but genital warts are curable. AIDS and genital herpes, herpes simplex virus and retrovirus, HIV virus, right? So, these two diseases are sexually transmitted disease which are not curable. There is no complete cure for these diseases. Okay. Next, which of the following statement is not true about Saheli? Now, Saheli is the miracle drug and we should be very proud on this, that CDRI, Central Drug Research Institute present in Lucknow, produce this drug Saheli. Saheli is a non-steroidal drug, one of a kind in whole world. It's a once a week pill, that means it is only be taken once in a week, and this have very less side effect because it is not hormonal. So, what we have to find is not true about Saheli. Okay. So, this is a non-steroidal preparation, this is completely true. Once a week pill, this is also completely true. Low contraceptive value, now this is wrong. If a pill is taken only once a week rather than other pills like eye pills which are taken daily for continued 21 days, that means Saheli is stronger than other pills. So, the low contraceptive value is completely wrong in this case, right? And has fewer side effect, this is also right because it is not hormonal based, that means it have very few side effect as compared to other drugs, right? So, the not correct, incorrect statement, not true about Saheli is it has low contraceptive value, right? Okay, now this is the most controversial question I have come across in this paper. Okay, understand this. A woman is unable to conceive even after many years of regular unprotected coitus. Okay, went to a specialized clinic. One complete examination, woman was found normal while the male partner was diagnosed with infertility. Now, infertility means either there are very few sperms, no sperms, or there are very less number. Now, less number or no sperms are two different problems, but in both the cases the male are considered infertile. If you can recall from the chapter 3rd, human reproduction, the NCERT clearly said that at least 60% of the sperm must have normal shape and size and 40% of them must show rigorous motility. And what is the concentration of sperm? Almost 100 million per ml, 100 million per ml. So, 60% means 60 million sperms, 40 million sperms must have rigorous motility. Then only a human male is considered as fertile. If it is lower than that, then the male partner is considered as infertile, right? So, infertility and was unable to inseminate the female due to low sperm count. Insemination. Now, there is a controversy here. What is, if a male is unable to inseminate because of fewer or lower sperm count, the insemination is not the problem, the sperm count is the problem, right? The male partner who have difficulty in insemination, that means entry of semen into the vagina, they must be suffered from erectile dysfunction, okay? But it's okay, it can be considered here, okay? Suggest the appropriate associated reproductive technology, the appropriate the technology which should apply here in this condition where male have the problem, what will be, okay? So it is ZIFT, zygote intrafallopian transfer. This is wrong. This is considered for the female problems, right? Now we have intrauterine insemination and we have artificial insemination. Both of them are the same thing, guys. Both of them are the same thing. Okay, gift is also not possible. This is also considered for the female problem. Now, what is the problem here? Artificial insemination is a broader term in which we inseminate a female. That means we enter the semen in the vagina artificially through injection or catheter or something like that. The artificial insemination is a broader term. IUI, that is intrauterine insemination, is the special kind of artificial insemination where the sperms are very, very few, that we have to inject the sperm directly into the uterus so it can reach to the ovum. Okay, so actually both of them are correct. Both of them are correct. Now we have to see the what they will consider as the right option. In the broader term, AI is the correct, but IUI is more correct here. When we are talking about the competition exam, IUI is more correct here. Both of them are correct, but IUI is more correct here. Okay, so we have to wait and see what will they consider as the right option. Maybe they will consider both 2 and 3 are right. There can be the bonus marks, or maybe God knows, but both of them are right, but IUI is more correct here. Okay. Now, the genotypes of the husband-wife is IA, IB and IA, IO among the blood groups of their children. How many different genotypes and phenotypes are possible? We have to know how many different genotype and phenotype. Phenotype is what? The blood group. Genotype is what? IA, IB, this kind of genetic makeup, right? So we have IA, IB, that is IA, IB, and we have IA, IO. So when we form the gamete we have is IA, IB, here IA, IO, right? This can be joined by this IA, IO, one possibility. Next possibility can be this one, IA, IA. Next possibility can be this one, that is IB, IA, and last possibility is this one, IB, IO. Okay, so you can see this here, the genetics is IA, IO, and the IO is the recessive one. So both of them will yield A blood group. Both of them will yield A blood group. This will yield in the AB blood group, and this will yield into the B blood group. So what we have is B, AB and A blood group. That means three phenotypes. But this is homozygous A and this is heterozygous A. So both of them have different genetics. So we have 4 type of genotypes in which 2 of them are making single phenotype, that is A. So phenotype are 3 but genotypes are 4. Okay. So 4 genotypes and 3 phenotypes. Second one is the correct option. Okay. I hope you understand this. Let's go to the next question. The classic example of point mutation is sickle cell anemia. We can take it right away. In the 6th chapter, molecular genetics, it has been given on in any other chapters or many chapters. It is also given in the biomolecules. It is also given. Sickle cell anemia is a point mutation where at the place of a polar amino acid, the valine comes in the place of glutamic acid at the 6th position of hemoglobin beta chain. At the place of glutamic acid, valine comes. Okay, so that is the and valine is a hydrophobic amino acid. So the complete folding changes. Okay, so sickle cell anemia is the point mutation. Point mutation is what? Where a single nucleotide change occurs, and because of that single nucleotide the amino acid changes. Okay. Let's go to the 38th question. Read the following statement. In a typical test cross, what a test cross is, we do test cross when we have to know the unknown genotype of any particular organism. See, we have two types of phenotype. One is recessive, one is dominant. Now the recessive have fixed genotype. Because it is recessive, that means it must be only homozygous, otherwise it can't express itself. But the dominant one can be heterozygous, can be homozygous. Right? Capital T small t also yield the tall plant. Capital T also yield the tall plant. But if I am having a tall plant, I cannot see whether it is homozygous or heterozygous. So to identify whether it is homozygous or heterozygous, we perform a test cross. And in test cross, we cross that particular plant with the homozygous recessive character of that plant. Okay. So in a typical test cross, an organism showing dominant phenotype, yes, is crossed with the recessive parent. This is completely right. Okay. Second, the ratio of organism showing dominant and recessive trait in F1 generation of the test cross indicate the organism of unknown genotype is homozygous. Now what this statement is saying, we have a ratio. Now we can only have a ratio if we have two types of genotype or two types of phenotype, right? If I have only one type of genotype, there is no ratio. At least two types of genotype or phenotype must be there, that means the ratio is present. But if there are two types of genotype, then the dominant one can't be homozygous. The dominant one can't be homozygous. Why? Let's see. Let's suppose the unknown genotype can be capital T, the unknown genotype can be small t, capital T, small t, right? If I cross this with this particular recessive parent, then all the plant will be capital T, small t, that means 100% are tall. So in homozygous condition, there is no ratio because every plant have dominant character. But if the parent is heterozygous, it can produce two type of gametes. So this will yield tall plant and this will yield dwarf. Here I can calculate the ratio because there are two possibilities, right? So if there is two possibility and the ratio will be always 1:1, in test cross there is always 1:1, or in dihybrid 1:1:1:1. Okay. So in test cross, if I have ratio, that means the parent is heterozygous, and if I don't get any ratio, that means the parent is homozygous. Okay. So this statement is wrong. So the correct statement is statement 1 is correct, statement 2 is wrong. So statement 1 is true, but statement 2 is false. Third option is the correct one. And statement 1 is true, but statement 2 is false. Okay. Let's go to the next question. Okay. Thalassemia and sickle cell anemia are due to the problem of globin molecule synthesis. Select the correct statement. Okay, thalassemia is a different disease, sickle cell anemia is a different disease. In sickle cell anemia, it is not the problem of amount of hemoglobin, it is the problem of beta chain folding. Due to faulty folding, it converts into the sickle cell RBC. Okay, because hemoglobin is not folded correctly. But in thalassemia, we have different kind of gene, A gene and B gene. Now A gene produce a little bit of hemoglobin, globulin protein, B gene produce little bit of hemoglobin, that is globulin protein. So if I have both A and B, I have this much of hemoglobin. If I have only A gene, I have this much of hemoglobin. So in thalassemia, the amount of hemoglobin is problem, while in sickle cell anemia, the folding of hemoglobin is the problem, not the amount. Now here we can calculate it, we can categorize it in quantitative and qualitative. Quantitative means the quantity matters, like our skin color. Okay, if I have more capital A, capital B and capital C gene, my skin color will be very dark. So the number of capital, that means dominant and recessive, here we can say the quantity of the dominant and recessive is the main thing here. Okay, so in thalassemia, I have quantitative problems because let's suppose I have two alleles of A and two alleles of B. I have 4 alleles. If 3 of them are dominant, I have this much, 75% of the hemoglobin. If two of them are dominant, I have 50% of hemoglobin. Okay, consider this like that. But in sickle cell anemia, it is not the problem. If the sickle cell gene is there, that means the faulty gene is there, all the hemoglobin will be folded differently. So it is qualitative. The quality of gene matters in the sickle cell anemia, while the quantity of gene is matters in the thalassemia. Okay. So we have to identify the correct statement. Sickle cell anemia is due to the quantitative problem of the globulin molecule. Now the quantitative is the problem. Sickle cell anemia is the qualitative problem. है ना? Quality of gene matters here. Both are due to qualitative defects? No. Both are due to quantitative defects? No. The thalassemia is due to less synthesis of globulin molecules. Less synthesis. Why? If two genes are faulty, 50% less. Like this. It's not only 50%. You can solve this by understanding in this manner. Okay. If both of all the alleles are there, 100% globulin protein. If some alleles are dominant, some are recessive or some are faulty, less synthesis of globulin protein. Okay. So the correct option is the fourth one, that is thalassemia is due to less synthesis of globulin protein. So let's go to the next question. Who amongst the following scientists had no contribution in the development of double helix model of the structure of DNA? Now double helix model is given by the Watson and Crick, but there are some scientists who are already have done work before Watson and Crick, like Wilkins and Franklin. They have done X-ray diffraction technique and they understand that there are two helix present in the DNA. Okay, Chargaff gave the famous Chargaff rule, which you used for in the calculation of adenine, thymine, cytosine and guanine. Okay, there is another question. The next question is based on the Chargaff rule. Messelson and Stahl have done the experiment that proves that semi-conservative method is the DNA replication method. Now, if you read the NCERT carefully, it is written in the NCERT, then after Watson and Crick gave the model, the Crick also said that because the DNA depends on complementary base pairing, we can also consider that it always replicate through semi-conservative method. Messelson and Stahl proved it with their experiment, the heavy nitrogen experiment, right? But the experiment of Messelson and Stahl comes after DNA double helix model is proposed. Okay, so they didn't do anything in respect of structure of DNA. So correct option is the third one. Okay. So next is the numerical based on the Chargaff rule. Very easy. It said in a DNA molecule cytosine is 28%. Calculate the percent of adenine. Okay. So what we can do is like this: C bonds with G with 3 hydrogen bonds and adenosine bond with thymine with 2 hydrogen bonds. Okay. If C is 28%, then G must also be 28%. Both 28 and 28 makes 56. So out of 100, 56 is cytosine and guanine. So rest 44 must be adenine and thymine. And if the 44 is adenine and thymine, then out of 44, 22 must be adenine and 22 must be thymine. Very simple. Okay. So the correct option is 22%. Right? Let's go to the next. Arrange below the steps of DNA fingerprinting in the sequence selection appropriate option. Okay, what we have to do is we have to choose the correct sequence for all these. Okay. Digestion of DNA by restriction endonuclease. Okay, this is the step. Autoradiography. This is the last step, completely last step. Okay. Blotting of DNA fragments. On the nitrocellulose paper, we blot the DNA, also known as Western blotting. When we already done separating and we have found our correct DNA. Okay. Isolation of DNA, this must be the first step because we can do anything with DNA. First we have to isolate it, right? And separation of DNA fragment. Now separation of DNA fragment can only be done after restriction of endonuclease digestion. So restriction endonuclease digestion is done and it breaks the DNA in many different parts. So this must be the second step. After separation, what the second step, separation is done, that is the third step. After separation, we have to blot it, we have to take it on the nitrocellulose membrane, and after that we have to do the autoradiography to see the DNA fragments in ethidium dibromide and ethidium bromide, sorry. So the correct option is D, A, E, C, B. D, A, E, C, B, third one. D, A, E, C, B. This is the correct option, right? Okay. Which is not true about the Big Bang theory? And a question is from evolution chapter. So Big Bang theory explains the origin of universe. Now this is the most accepted theory, Big Bang theory. This is given by AB scientist. As universe expanded, the temperature came down. This is completely true. When anything expands, it loses its energy in expanding. So the temperature came down. This is also correct. Hydrogen and helium gas formed first, but here it is written before the explosion. Without explosion, how can the gases form? Gases can only be formed after the explosion, right? It is saying before. So this is not correct. That means the answer will be 3. Okay. And gases condense under gravitation formed galaxy. This is the right one. So only incorrect one is this one. And because of this, right? Let's go to the next question. According to Hugo de Vries, speciation. Hugo de Vries is a scientist who gave the mutation theory, and he is also the member of 3 scientists, not a team. They are working independently in different countries, but he is one of the scientists who rediscovered Mendelism in 1900. Rediscovery of Mendelism. Hugo de Vries was working in the on the evening primrose. Okay, and he also gives the mutation theory. Now, what is the mutation theory? Single step, large variation. Single step, large variation. Direct change in the DNA, and this change will be hereditary. It will change the DNA, and the DNA carries the genetic information. That's why these changes will go generation after generation. Okay. So accumulation of small variation, no, this is natural selection. Intraspecific breeding, there is nothing about the breeding here. Interspecific breeding, also no. Single step, large mutation. The correct option is the 4th one. Okay. Now let's go for the next question, that is I guess 45. Identify the correct order of stages of human evolution. We have to identify which comes first and which comes later. Okay. So first we have Dryopithecus, then we have Ramapithecus, then we have Australopithecus, then we have Homo habilis, then we have Homo erectus, then we have Neanderthals, Cro-Magnon, and Homo sapiens. Okay, this is the order of human evolution. So let's see, Dryopithecus, E first. This is the correct option. Okay, everything is starting from another thing. E first. Okay, so after E, C. After Dryopithecus, Australopithecus. So after Dryopithecus, Australopithecus. Then after Australopithecus, Homo habilis. Then after Homo habilis, Homo erectus. And after Homo erectus, Neanderthal man. Right? Okay. Let's go to the next question. Pollen grains are well preserved as fossil because of the presence of sporopollenin. This is the direct question. You know that the pollen grains have very tough wall in terms of exine. Exine is made up of sporopollenin. The sporopollenin is the strongest organic metal known, organic matter, right? It can't be degraded from any enzyme. There are no enzymes to degrade it. It can be destroyed by alkali. It can be destroyed by the acids. Nothing works on it, and that's why they are preserved as fossils. Okay, and who produce this sporopollenin? Tapetum, with the help of Yubish bodies, produces sporopollenin, and this sporopollenin forms the exine of the pollen grain. Right? The sporopollenin is the direct question. The causative agent of pneumonia. Pneumonia is Haemophilus influenza. It is a bacterial disease. है ना? Either Haemophilus influenza or Streptococcus, also Staphylococcus pneumoniae. These two bacteria are the causative agent of pneumonia. Okay. Select the option that contains only secondary lymphoid organs. Now, what are secondary lymphoid organs? Let's understand the concept first. We have two type of lymphoid organ. One is primary lymphoid organ and another is secondary lymphoid organ. The primary lymphoid organs are those organs which produces the cells. Okay, they are the production factories of the cell. While the secondary lymphoid organs are those places where these cells go and fight. Okay, so these cells are produced. What kind of cells? WBCs, T cell, B cells, right? So this location where these cells formed or mature are known as primary lymphoid organ, and after maturation they goes to the secondary lymphoid organ to fight of the causative agent or bacteria or any other microorganisms, right? So we have only two primary lymphoid organs, one is bone marrow and another one is thymus gland. So bone marrow, you know, bone marrow is present in the longer bones. The bone marrow have hematopoietic stem cell which produces all type of blood cells. One of them also produces lymphocytes. Now if the lymphocytes are matured in bone marrow, then they are termed as B lymphocytes. But if they migrate and go in the thymus and are matured under the influence of thymosin hormone, that means it will form T lymphocyte. So in NCERT it is written that the bone marrow and thymus provide micro environments for the maturation or formation of B lymphocyte and T lymphocyte. Now what are secondary lymphoid organs? Secondary lymphoid organs are those places where these B and T lymphocytes will migrate and action. Okay, so what we have lymph nodes, the Walder's rings are present in there, and every opening we have tonsils. Tonsils are nothing but the lymphoid tissues. Okay, so lymph nodes, lymph vessels, Peyer's patches present in the near the intestine. Okay, MALT, mucosal associated lymphoid tissue, more than almost 50% the lymphoid tissue of the body. Okay, so all these are secondary lymphoid tissues, right? So let's know, select the option that contains only secondary lymphoid organ. So spleen, tonsils, thymus. Thymus is the primary. This is wrong. Spleen is also the secondary lymphoid organ. Peyer's patches correct, tonsils correct, but bone marrow is wrong. Lymph nodes, appendix, tonsils, these all are correct. MALT can also be written. Lymph nodes can also be written. All these are secondary. Okay? Here tonsil, thymus is the primary one. Okay, the correct option is the second one. Only secondary lymphoid organs, right? Let's go to the next question. Identify the biological response modifier substance which activate immune system and helps in destroying the tumor. Now tumor is cancerous cells, those cells which rebels against the body. They lose the property to stop their dividing process. They lose the property of contact inhibition. When a cell divides, when it touches the another cell, it comes in the contact with another cell physically, it knows that there is no place, no space now. So I have to stop division. But a cancerous cell lose its this possibility, and it kept on dividing and hence made a tumor. Now our immune system can identify these cancer cells and destroy them. It is said that we produce cancer cell daily, but our immune system can locate them and destroy them before they make a tumor. If our immune system is failed to do that, then cancer can be caused. Now interferons are some chemicals which are produced by the cells which can help the immune system to destroy these cancer cells. Okay, so biological response modifier or substance, this is alpha interferon. Alpha lactalbumin is something which is present in the milk, and the rosy cow, if you remember, it have alpha lactalbumin. It is given in the biotechnology, right? Serotonin is happy hormone. 90% of the serotonin is produced in our GIT tract by argentephene cells, and 10% of them is produced by pineal gland. Okay, and histamine is a vasodilator. So it is a vasodilator. So what is the correct answer here? Alpha interferon. Okay. Next, the conversion of milk into curd by lactic acid bacteria increases the nutrition value by producing vitamin B12. This is the direct question given in the NCERT. Okay. Next question, BOD, biological oxygen demand is polluted water is estimated by measuring the amount of. Now let's understand what is BOD. Okay. Suppose there is a water body. In the water body, organic matter, that means organic waste, biodegradable waste is present, right? Now the microorganisms can feed on that biodegradable waste because they are decomposers. In doing so, they require oxygen because they are also require oxygen, they are aerobic bacteria, okay? Now, if the organic matter is more, then bacteria require more oxygen to degrade it. Understand this. If you are 10 people, you can eat a certain amount of food, but there is this much amount of food, then you require more than 10 people, right? In the same case, if the organic matter is high, then the demand of oxygen by the microorganism is also high because they require more oxygen in utilizing the organic matter, and the organic matter is something which polluted water. So more the pollution in the water means more organic matter here, which means more oxygen demand by microorganism to degrade this organic matter. So biological oxygen demand, है ना, biological oxygen demand, biological, why? Because living organisms demand what? Oxygen. That's why it is called as biological oxygen demand. So it is the amount of oxygen required by the microorganism to degrade the organic matter present in a particular water body. Okay, so it is oxygen consumption. It is estimated by the amount of oxygen consumption. More oxygen consumption means bacteria need more oxygen, that means more organic matter must be there, that means more pollution must be there. Okay. Let's go to the next next question. The given below are two statements. Baculovirus are pathogen that attack nematodes. Now this is wrong statement. Baculoviruses are the viruses which attacks on specific genus or specific range of insects. They attack on insects, means arthropods. They don't attack nematodes. Thus this statement is completely wrong. Baculovirus are used as biological control agents, ecologically sensitive areas, as they species specific in their action. Now this is completely right. It is also written in the NCERT. If I have to protect my plant from insect, but if I spray insecticide, the insecticide will kill all type of insects, even honeybees, butterflies, which are actually good for me because they are doing pollination. The baculovirus only attacks to the specific genus or specific species of the insect. So the ecologically sensitive area, that means this area is very sensitive, little bit of fluctuation can be result in the endangering of some species. So here baculoviruses are used. So statement 1 is wrong, but statement 2 is the correct one. So statement 1 is false and statement 2 is true. The correct option is the 4th one. Okay. Let's go to the next question. The technique to alter the chemistry or genetic material of DNA and RNA and introduce these in the host organism and change the phenotype of the host is genetic engineering. Okay. So biotechnology process chapter, on the first page it has been written that there are two main aspects of biotechnology. First is genetic engineering. In genetic engineering, we alter the chemistry of DNA and RNA and introduce them into the host, and because the change is change occurs in the DNA and RNA, this changed DNA and RNA is result into the different kind of phenotype. Okay. And second one is bioprocess engineering. Bioprocess engineering is all about creating a microbial free environment so that we can grow our choice of microorganism on a large scale for the production of large scale molecules, right? So bioprocess engineering is completely after the formation of genetically modified organism. Okay, so this is genetic engineering. This is the direct question, genetic engineering, right? Let's go to the 54th question. So given below are two statements. First is restriction enzyme BAM-H1 has its recognition site on tetracycline resistance. This is completely right. here we have ampicillin resistance, here we have tetracycline resistance, and BAM H1 and SAL1. These restriction endonucleases are present in here. So if I use BAM H1, the tetracycline resistant gene is destroyed. Okay, this is complete tetracycline resistant gene from here to here, and this is the site of BAM H1. So if I use the restriction endonuclease BAM H1, it will break it. It will break it. So the DNA is compromised now. That gene which create something by which the bacteria is protected in the tetracycline region. Tetracycline is a kind of antibiotic. Okay, that gene is destroyed because of BAM H1. Because of BAM H1, that gene is destroyed. So bacteria can't grow in the tetracycline region. Okay, so first is the correct statement. BAM H1 has its recognition site in tetracycline region in PBr 322. PBr 322 is the artificial plasmid, first form artificial plasmid done or formed by Bolivar and Rodriguez BR in 322 attempt. Okay, and E. coli having pre-BR 322 with desired DNA, if inserted at BAM H1 site, can grow in the medium containing tetracycline. This is incorrect because tetracycline gene is compromised if I am using BAM H1 site. Okay, so this statement is wrong, this statement is right. So the correct option is statement 1 is true and the statement 2 is false. Let's go to the next question. Okay, match the following. Now, visualization of DNA in gel electrophoresis. This can be done by ethidium bromide. Ethidium bromide, we use ethidium bromide. The ethidium bromide attaches with the DNA molecules, and then when we put that DNA molecule in the UV spectrophotometer, then ethidium bromide is produced as bright orange color. So we can see the DNA with the help of ethidium bromide under UV spectrophotometer. Okay. Precipitation of DNA, chilled ethanol. Chilled ethanol is responsible for the precipitation of DNA. Breaking the cell wall. Now bacterial cell wall can be break by lysozyme enzyme. Our saliva and tears have lysozyme and they can break the cell wall of bacteria. And the breaking the cell wall of fungus, the fungus cell wall is made up of chitin, so chitinase is the enzyme. Chitinase is the enzyme. So A fourth here, A fourth and B third. So first is the correct option in this case. Okay, just 4 more question, guys. Okay, let's do it. So the human protein obtained from transgenic animal used to treat emphysema. Emphysema is also known as smoker's disease. This disease is given to you in the class 11th chapter, breathing and exchange of gases. What happens in emphysema? The smoking produces, you can say that, let's understand it from the basics. What we have, our alveoli have these kind of walls. Our alveoli have these kinds of walls. It is completely not smooth. It is like this. Now these walls are made up of proteins. These walls are made up of proteins, and trypsin is an enzyme which break these walls, Okay, so we don't want trypsin to break these walls. That's why our body have alpha 1 antitrypsin. The word itself says anti of trypsin. So this stop the trypsin to cause this damage. Now in the patients who are suffering from emphysema, the alpha 1 antitrypsin does not form. And if the alpha 1 antitrypsin does not form, then trypsin is completely free to degrade all these walls. So this alveoli is converted into like this. And due to the reduced surface area, the patient of emphysema can can inhale sufficient amount of air, but the exchange is not up to the mark. Okay, so the alpha 1 antitrypsin is the chemical which is used to treat emphysema. When alpha 1 antitrypsin enters in the body, it will stop the trypsin for degrading these kind of walls. Okay, and our alveoli will be protected, right? Let's go to the next one. Okay, insulin, you know, it is a proteinaceous hormone and it lowers the blood glucose level. Alpha lactalbumin, I already told you, alpha 1 lactalbumin and beta lactalbumin, these are present in the mother's milk, okay, and these are very, very crucial for baby's development. Next question, given below are the sequence of event in the production of human insulin by Eli Lilly. Now the human insulin is produced in the inactive form proinsulin. What proinsulin is? It has A chain, then it has C chain and it has B chain. Now A chain and B chain are linked together by disulfide bond. This C chain needs to be removed and this is the actual insulin. This is proinsulin, which is inactive form. Now the insulin have A, B and C chain and our gene are such like it will produce A, C and B chain. So we cannot change here. But if we are considering to cultivate insulin in artificial environment or through the GMOs, the whole gene must be inserted. Now if the whole gene must be inserted, then proinsulin will form, and because the insulin gene is foreign to that bacteria, the process from proinsulin to form insulin is not present in the bacteria. So what Eli Lilly does, this company broke the DNA and put A gene in one type of bacteria and put B gene into the another type of bacteria. So this bacteria produces only A chain, this bacteria produces only B chain, and then after that, after isolation, they artificially make them bonded with disulfide bridge and the insulin is formed. That is called as humulin. Okay, so it's a very great study. So what are the correct sequence? We have to find the correct sequence here. So preparation of two DNA sequence corresponding to the chain A and B in human insulin. Okay, this production of chain A and chain B separately. Okay, introduction of chains in the plasmid of E. coli. Okay, extraction of chain A and B and combining them to create disulfide bond. Okay, this is the last option. Okay, this is the last option, right? So what first we have to do is production of two DNA sequence corresponding to chain A and B. First we have to take out the DNA of A chain only and take out the DNA of B chain only. So this one will be the first. After that we have to introduce these chains in E. coli. So the E. coli will become a genetically modified organism, a transgenic bacteria. After that we have the production of A and B chain separately, and after that we can isolate them and we can make disulfide bridge. Okay, so the correct option is A, C, B, D. A, C, B, D. A, C, B, D. The second one is the correct option here. Okay. Now, here are two statements. Adenosine deaminase is crucial for immune system to function. Now, adenosine deaminase is a very important enzyme which is helpful in formation of immune system and development of immune system. If ADA deficits and if ADA deficiency present in any human being, then their immune system cannot function properly. Okay. So, adenosine deaminase deficiency can be cured only by bone marrow transplantation. Only by bone marrow transplantation is wrong. We can also inject adenosine deaminase because this is an enzyme. Okay, we can synthesize it in laboratory by using the E. coli or some other bacteria. ADA is an enzyme, so I can also introduce the enzyme intravenously in the body if I want to. Another thing, I can do the bone marrow transplant, second option, or I can also change the cells present in bone marrow of the host by adding the ADA gene in them. So there are a number of procedures. Bone marrow transplantation is the complete and permanent cure, but it is not the only cure. Okay, so this statement is wrong. This statement is right. This statement is wrong. So statement 1 is true and the statement second is false. This is the correct option, right? And there are last two questions. So question number 59, which of the following is not correct with reference to exponential growth? What is exponential growth? J shape. J shape. There is no limit of resources, there is no carrying capacity, organism can reproduce in their fullest extent. That kind of growth is known as exponential growth. Okay. So not correct. Resources are limited. That is not correct. In exponential growth, if the resources are limited, the exponential growth is not possible. Only logistic growth is possible. Right? Population grows in a geometric fashion. This is completely right. What is geometric fashion? 2, 4, 8, 16, just doubling every time. 2 to the power n. A stationary phase is never reached. This is also right because we are going like this upwards. So we have no stationary phase. Stationary phase can only be seen in the logistic growth curve. Right? And the population grows beyond carrying capacity. This is also right. If this is the carrying capacity, this is the logical growth. This is the logical growth. But exponential growth crosses the carrying capacity. Right? So the only not correct is the first one. That is resources are limited. Okay? And this is the last question, guys. The graph present age pyramids of the population. We have to identify which one is this. So if we have this kind of pyramid where the pre-reproductive and reproductive phase both are same, then this is called as stable. This is called as stable. This is called decline. Decline we can identify very easily, right? And this is called as what we can say expanding. Expanding, right? The correct option is the third one. Okay. So I hope all the questions are clear to you. Please comment below how much score did you guys get in according to this answer key, and let me know if you have any doubt in the comment section. I will be happy to help you guys wherever or whenever and however I can. And please be confident for your NEET exam. The NEET exam is going on, so we don't have to lose faith. We don't have to lose the confidence. There is still time. We can do it. You can do it. Okay. So I'll see you in the next video. Till then, take care, guys.